Welcome to On the Edge of Breakthrough, where hope meets innovation in the fight against cancer.
Hi, I'm Dr. Monty Pal. I'm a medical oncologist at City of Hope, and I'm really excited to bring to you On the Edge of Breakthrough, Voices of Cancer Research. We're going to be bringing some of our best researchers to the table to discuss exciting findings from their laboratories, from their research programs, and really get some great perspectives on what their plans are for the future. Welcome back, everyone. I have no doubt that you've probably come across my guest today, Dr. Charles Brenner, our chair in diabetes and cancer metabolism on social media. He's done a great job of vocalizing his research in that setting, and I can't tell you how excited I am to sit down with him and finally have a chance to chat with you about this amazing work that you're doing, really at this novel intersection, right?
Yeah, it's exciting to be here at City of Hope. And City of Hope is unique in having established a Department of Diabetes and Cancer Metabolism right at the interface of the Cancer Center, our Comprehensive Cancer Center, and the Arthur Riggs Diabetes and Metabolism Research Institute.
And this is really the first of its kind, am I right? I haven't heard of a lot of other institutions that have really put such an emphasis on understanding the continuum, perhaps, between these two diseases.
Right. We're right at the interface, and given that there is an epidemic of diabetes across the world, we see that it's driving cancer incidence and severity at a lot of different organ sites.
Now, we're going to dive into your research indeed in just a moment here, but I think a lot of people may not know about this rich history that City of Hope has in the space of diabetes research. Can you kind of walk us back a little bit and tell us about how City of Hope had its roots really in that space?
Early on, as City of Hope became a research center, there were very important discoveries that were made in diabetology. So hemoglobin A1C as a biomarker of elevated glucose was discovered here. The role of insulin in getting circulating blood glucose into skeletal muscle was discovered here. And then you can't, ignore the first product of recombinant DNA technology, right? Which was human insulin, the technology of which was developed here by Arthur Riggs and his co-workers. This was licensed by Genentech and became a huge global product.
It really is the whole spectrum of diabetes management, right? Because you think about diagnosis using hemoglobin A1C, you think about the day-to-day management of patients with diabetes using insulin. I mean, it really seems as though City of Hope has been a focal point through all that. And of course, along the way, we've developed such an impressive body of work in the area of cancer. And I do see in the literature, maybe over the past decade or so, more and more interest in really sort of understanding this intersection, perhaps with not the same laser focus that we've got over here. But in very broad terms, can you tell me why and how, and we'll get into the weeds later, there's some connections between the two disorders?
I got interested in fatty liver disease. It's now called MASLD, or metabolic dysfunction-associated steatotic liver disease. And that's really a key driver of both type 2 diabetes and hepatocellular carcinoma. And there's increased incidence of both type 2 diabetes and hepatocellular carcinoma. There's transcription factors that are involved in the development of fatty liver disease. And basically, our hepatocytes and our pancreatic beta cells haven't had time to evolve to the new environment that we're in which with a few dollars, you can have 2,000, 3,000, 4,000 calories every day, right? And lesser demand on our bodies to hunt for food, to grow our own food, to harvest our own food. And so there's a mismatch between our energy availability and our energy intake and our energy expenditure. And it leads to development of adiposity inside of cells, in adipose cells themselves, and in ways that dysregulate the body and lead to higher burden of cancer.
Yeah, I think this is a totally sort of new way of thinking, this whole concept. And I saw it, described in some of your work as overnutrition. Now I want you to take me back. So you started your career getting a BA in biology at Wesleyan, and then were off to Stanford for your PhD. Where was it in that time frame that you got interested in this body of research?
It was probably about 45 years ago when I decided to spend the summer after my sophomore year in college as a research assistant for two of my favorite faculty members. One of them was a brilliant intellectual historian. The other was a brilliant Drosophila molecular biologist. The brilliant intellectual historian was trying to write a book on Sigmund Freud. It took him another 10 or 15 years to complete that body of work. And essentially, being an assistant to him meant that I was going to the library to try to find in which book, Sigmund Freud said one thing or another. It wasn't any new original research on my part. I went into the biology professor's office and he said, "We know that XY flies are male, but we don't know why they're male. We don't know whether it's dosage of information on the X chromosome or something on the Y chromosome that masculinizes them." And he said, "We just don't know, but we need you to go and try to clone some DNA from the Y chromosome and figure out what is encoded there and figure out how this works." And I just thought, wow, that is amazing that you can just go in, you can solve a very important problem with your own labor and with your own intellect. So I could never go back after that.
It's so funny because the Y chromosome versus X chromosome sort of conflict comes up in the context of cancer a lot. And I've seen a lot of papers in Nature and Science recently around Y chromosome kind of driving diseases like bladder cancer. It's interesting how those fundamental questions still sort of linger on in present day.
Right. And we gave credit to Dr. Arthur Riggs for doing recombinant human insulin and bringing in the licensing money that built a lot of buildings around here. Arthur Riggs actually, when he was a new faculty member, wanted to figure out how Xin activation works and also discovered some key aspects of DNA methylation. So it's a remarkable set of discoveries that are linked here to City Folk as well.
So that was the initial sort of focal point in college, but how did you sort of transition to your PhD work and what did you focus on then?
Well, it's funny. So I'm 21 years old, graduating from Wesleyan University. So I'd taken the GREs to go to grad school, and I had like 99th percentile in verbal, quantitative, and analytical, but I didn't apply to graduate school because nobody in my family had a doctorate. We didn't have any MDs or any PhDs in my family. I had the charming notion that you get a college education to not be a burden to your parents. So what I decided to do is go to the San Francisco Bay Area and get a job in biotech. And so with my new bachelor's degree in biology from Wesleyan, I went to work a couple of years at Chiron Corporation in Emeryville, California, when it was a tiny company. And then a couple more years at DNAX Research Institute in Palo Alto. It's not quite a Stanford spinoff, but almost. And it was wholly owned by Shearing Plough, in fact. But it was a kind of a basic research institute that was run in the private sector. And I had enormous, opportunities in those industry stints. 88 is when I started graduate school, and I went to Stanford. I worked in the biochemistry department at Stanford, PhD in cancer biology, and essentially I purified an enzyme that is involved in pro-hormone processing. You probably know about PCSK9, right? Which is
cardiovascular drug,
huge drug for control of cholesterol. That PCSK9, it refers to prohormone convertase, subtlysin, kexin superfamily. Those enzymes are all related to the enzyme that I purified in graduate school, KEX2 protease. So I did the first purification of an enzyme in that class.
That's so cool. And I should warn our audience, we're going to have a lot of complicated acronyms today.
Yeah, so we're kind of nerding out here a little bit, but we'll try to keep it simple.
It's funny you mentioned working at Chiron. I treat kidney cancers, and they've, of course, played a huge role in the initial development and commercialization of IL-2 and agents like it. We've certainly had some interesting intersects through our careers.
Yes.
Great project at Stanford. Where'd you go from there?
So it was effectively 10 years in California, about 5 years in biotech and 5 years getting my PhD. Then flew back to Boston, or actually drove back to Boston in 93, and I did my postdoc at Brandeis University. I had a nice background completing graduate school because based upon my work in biotech, I had experience in yeast genetics and yeast molecular biology, and then in enzymology in graduate school. And I got more and more interested in how enzymes work. And I decided to train in an X-ray crystallography lab. So in a lab that does protein structures, specifically on enzymes. That lab was run by Greg Petsko and Dagmar Ringe. Greg's a National Academy member. And I was there for between 2 and 3 years, learned X-ray crystallography, got a very nice publication. And then 1996, so 93 to 96, I was a postdoc at Brandeis. And then I got my first academic job, independent job in Philly at Thomas Jefferson University.
That's awesome. And at Thomas Jefferson, is that where you started the focus thereafter on diabetes research?
I was just a biochemist nerd working on my favorite classes of enzymes and proteins. And the thing that emerged out of my postdoc was a superfamily of nucleotide binding proteins. So, RAS, and there's a lot of proteins that interact with nucleotides. Nucleotides are the building blocks of RNA and DNA. But then ATP is a nucleotide, NAD, which is what I'm mostly known for, is a type of a nucleotide. And there's professional classes of proteins that interact with nucleotides. Mm-hmm. And so I was the guy that characterized nucleotide binding proteins. And in the course of doing that work at Thomas Jefferson University, and then in my next job at Dartmouth, we ran into the mother of all, energy producing nucleotides, which is NAD, nicotinamide adenine dinucleotide. So it's the central coenzyme of metabolism. It carries high energy electrons that allow us to live. In the course of working on NAD, metabolizing enzymes, I discovered a new vitamin pathway, a new way that cells can make NAD through this molecule called nicotinamide riboside.
Awesome. And this really sort of lends itself to your reputation as really, I would say, an internationally recognized expert on NAD and its role in metabolism and so forth. So that really began during your time then at Dartmouth, is that right? Or
Yeah, it was kind of a transition between Jefferson. At the end of my 7 years in Philly, I was already working on NAD-dependent enzymes. But when I got to Dartmouth, I was, as I normally do, nerding out on an enzyme. I liked it because it had not one, but two active sites.
Okay.
And I'm trying to figure out how those two active sites work together. You could open up your favorite textbook and it would say, this is how NAD is made. So it would have a bunch of arrows coming from tryptophan, which is an amino acid. And it would have some arrows. This is if you start with vitamin B3, right?
Okay.
And it would describe two forms of vitamin B3, one niacin, nicotinic acid, and the other is called nicotinamide. And so there's various arrows and there's various genes and enzymes that are responsible for converting known vitamins to this NAD coenzyme. And I just asked a kind of a radical question, which is this true? And even if it's true, is it complete? Because I can draw other ways of making NAD. In fact, I know that in bacteria, there are other ways of making NAD. Some bacteria can make NAD from aspartic acid. Some bacteria can make NAD from a nucleoside called nicotinamide riboside. And so I realized that there was an experiment that I could do that would test whether the received wisdom in the textbooks was true and correct. Pavel Bieganovsky and I did that experiment. And in 2004, we had a Cell paper on discovery of NR as a vitamin and discovery of the nicotinamide riboside kinase genes, which are found all the way from yeast to humans. A few years later, there was so much interest in NAD that this technology became commercialized, and it actually produced something called the NAD boosting industry, where there's 125 clinical trials of nicotinamide riboside. There's other NAD boosting compounds that are being tested in diseases and conditions.
And what do you think of those?
Nicotinamide riboside, there are 8 positive clinical trials showing anti-inflammatory activity. There's absolutely a use case for nicotinamide riboside. One of the problems in the field, in the NAD space, where they say you take this and take resveratrol and take some weird other compounds and you won't age and it'll activate your sirtuin genes and this kind of thing. And so there's a bunch of stuff that is not evidence-based in the space, but nicotinamide riboside biology is real. And, there's a large trial that is being completed in Norway this year in Parkinson's. There was a pretty large trial in peripheral artery disease where it was a placebo-controlled trial. The people with peripheral artery disease on nicotinamide riboside were able to improve their walking speed.
Okay.
In the placebo group, they decreased their walking speed over the course of the trial because they have a debilitating illness. And so there's absolutely use cases for NR, but there's also, some hype in the NED space that I try to tamp down.
Just as in any sort of cancer therapeutic trial really requires that very sort of specific and regimented study.
I mean, it's got to be placebo-controlled.
You've kind of walked us through transition through Dartmouth, and you're, of course, the founding director of a department here at City of Hope.
Yes.
But you also started a new department during your time at Iowa. Is that right?
I became the head of a esteemed department that has a long history. In 2009, I was recruited to University of Iowa to be the head of biochemistry. That was great. I mean, it was an interesting department because we're one of the 5 basic science departments in a medical school with, I guess, 22 clinical departments, something like that. So there's a big dean's meeting and all that. But in addition to that, we taught biochemistry to dental students, to pharmacy students, to undergrads. Okay. We had an undergraduate major. And in fact, I assigned myself metabolism. I taught metabolism to undergraduates for 11 years.
Were you a tough teacher?
It made me so much better at understanding what I do and understanding what's known and what's unknown. Yeah. I don't think I was a tough teacher. I loved it when my top 1 or 2% of students would ask me really cutting edge questions like, why is fructose more lipogenic than glucose?
Okay.
And there are 2 answers that are in the textbooks, but just in the last year or 2, we think we've discovered a 3rd answer by doing research. This takes me back to your first question of how'd you fall in love with science? It's the unanswered questions that you can go in and try to answer.
There's so much, I would say, true to the fact that getting back to the basics, right, and really understanding the fundamentals is key. Mm-hmm. I'm coaching my son's Science Olympiad team, right, and getting back to my first anatomy textbooks and physiology textbooks. So this is for, middle school and high school kids, but depth of understanding you have after you sort of reinsert yourself in those fundamental studies is so valuable, isn't it? So that was your time at Iowa. How did we pull you over here to City of Hope?
Got a phone call from Debbie Thurmond. Debbie Thurmond got her PhD at the University of Iowa in the biochemistry department, went off to University of Indiana. I tried to recruit her from Indiana to University of Iowa. She came here. She was recruited by Arthur Riggs to be a department chair. She had this great opportunity that she told me about. She said, we're known for cancer. We're known for diabetes and metabolism. We want to create an interface department. We know you have a PhD in cancer biology from Stanford. We know you do NAD metabolism. I'd never been to City of Hope before, but I visited 2 or 3 times. I moved during the pandemic. It's been wonderful to come here and to make connections and collaborations across the Cancer Center, as well as across the Diabetes and Metabolism Research Institute. So there's about 31 or 32 faculty groups in diabetes and metabolism. There's about 70 or so research groups in the Cancer Center, 100 or so faculty in the unit here that's called the Beckman Research Institute. And John Carpton is the scientific director of the Beckman Research Institute and the director of the Cancer Center. We didn't want diabetes and metabolism to exist in a kind of parallel play situation with the Cancer Center without really a lot of cross-fertilization. So my role in the Cancer Center is I co-direct what's called Molecular and Cellular Biology of Cancer. It's kind of the basic science program in the Cancer Center. And I really try to build metabolism research into that basic science program because metabolism goes into therapeutics. Right. Metabolism goes into cancer immunology. Metabolism goes into population sciences. There's nothing that you can you could try to play a game with me and try to ask me of something that is unrelated to metabolism. I don't think you can succeed. There's a metabolic component of every thought that you're thinking, everything that you've ever done. And so I try to infuse metabolism into the cancer center. And then at the same time, I'm the chair of a small department that reports into Debbie Thurman in the Diabetes and Metabolism Research Institute. So we're growing that interface here.
So you've been in a variety of settings that we've highlighted, cancer center now, traditional university,
Yes.
industry. What's the difference being at a place like ours versus the others?
It has a biotech-like component because of the tremendous accomplishments of people like Arthur Riggs and the fact that the shared resources here include manufacturing resources. So there's small molecule synthesis, there's drug screening, there's biological production capability here. Clinical trials are very meaningful and very substantive here, as you can explain to the viewers. There's a lot of cancer centers where, in order to be a comprehensive cancer center, you've got to take things from the bench to the bedside, and you've got to enroll some of your patients in your catchment area into clinical trials. But the number of patients that are enrolled in clinical trials here essentially exceeds all national expectations and has really improved standard of care for people globally. And so that's an exciting thing about being at City of Hope. I kind of miss having undergraduates and teaching metabolism to undergraduates, but of course we have a PhD program here. We have postdocs. We have outstanding colleagues in medicine, such as yourself. So it's been a very nice transition.
And I'll tell you what, I'll come to your office so you can teach me some basic biochem again. I could definitely take a...
We could do the malate aspartate shuttle, believe it or not. So, we found a disease where it inactivates something that you probably haven't thought about since first year in medical school.
Well, why don't we talk about that? Let's first dive into this really, I think, timely foray that your laboratory is focused on, which was fatty liver, or MASLD, as you previously described it. Tell me about what your lab is focused on in that regard.
Think about breast cancer, genetics. Breast cancer is so common in women, ballpark 1 in 10 in her lifetime. And so if you were to try to draw the genetics of a breast cancer in most families, you know how you draw these pedigrees where there's a square for dad,
sure,
there's a circle for mom, right? And then you draw a line. I wish we had a whiteboard. You draw a line between the square and the circle, and then you can draw their kids. And then if somebody ends up with a disease, you color in their circle or square. So if you look at most families, just 10% of the women over their lifetime will have a diagnosis of breast cancer. It's so common environmentally and sporadically that it's very hard to see the genetic basis for it. But Mary Claire King did something very clever, which is she said, I want to look at families in which there's men with breast cancer. So you ignore most of the families where men never get breast cancer. And then if you see these families where there's a square that's filled in, a guy that has breast cancer, despite the fact that he has lower levels of the hormones that we call female hormones, and he has less breast tissue. If he gets breast cancer, there's a very powerful gene at the root of that breast cancer. And that's where the BRCA1 and BRCA2 genes were discovered in families where men have breast cancer. So if you look at fatty liver disease, MASLD is the technical term now, you generally find it in people with overweight, but not exclusively.
Mm-hmm.
So there is such a thing as lean MASLD, people that have our body mass index, but if you do imaging of the liver, you see fat in the liver. So those lean MASLD syndromes, that means that there's something genetically that's really powerfully driving this process, because you shouldn't be accumulating fat in hepatocytes. If a person is really skinny, overnight, any fat that they have deposited in their hepatocytes should be used to burn to make ATP.
Sure.
Right? So I got really, really interested in a lean MASLD syndrome. It's a rare disease. It's called citrin deficiency. There's only one mutation that is required in humans to make it. Believe it or not, gene name is called SLC25A13. Believe it or not, there's more people globally with citrin deficiency than with cystic fibrosis. There's more people with CD than CF, even though you never hear about citrin deficiency in the United States.
You mentioned that to me ahead of the podcast. It just got me thinking. I mean, I sit in my clinic all day looking at CT scans, and it bewilders me the amount of fatty change that I see in livers, just as you mentioned, of young patients who are thin, terrific body habitus and so forth. It's really sort of incongruous with the levels of obesity that you'd anticipate with fatty liver.
Right. Well, one of the things that we discovered is that there's a transcription factor that gets activated in this disease of citrin deficiency, and there's a small molecule that activates it that drives the fatty liver disease. Another thing that's in our next body of work is that there's a very common variant, a very common polymorphism in the transcription factor. So the people that are lean with fatty liver, we think are much more likely to have a rare variant in a particular transcription factor. And so there's a lot of things that we're doing that we're going to be able to take from our mouse experiments and our laboratory experiments into people, where we'll be screening for a particular mutation and determining what increased risk that gives a person for type 2 diabetes and for hepatocellular carcinoma, as well as other types of cancer.
Fascinating. And how about on the therapeutic continuum for that? What might you say could potentially address this issue?
There's sort of 3 different therapeutic implications of the work that we've done on citrin deficiency. The adults, like I said, have lean MASLD. So they're skinny, but they have a fatty liver. We have 2 or 3 drug targets to potentially relieve the liver, or to mobilize the fat from the liver so that the liver doesn't retain these lipid droplets and will activate that. And we're going to be testing those in our mouse model. A second therapeutic angle that has come out of this disease is that this disease is a type of a urea cycle disorder. This is another thing that probably takes you back to M1 medical school, right? Yeah. So, but it's not, there's high levels of a compound called citrulline in these patients, and but yet they don't have a mutation in ASS1, which is usually the cause of that urea cycle disorder. They have a different mutation. But we found drug targets that we think will relieve the complications of urea cycle disorder in this population. And then the third thing is that these patients are really interesting because they don't like sweets. This is one of the things that got me really interested in the disease, is that it's a lean MASLD syndrome. So that reminded me of men with breast cancer. There's got to be some really powerful gene dysregulation involved in storage of fat in a skinny person or a skinny mouse. And we discovered that. But then the other thing that made me really interested in the disease is they don't like sweets. And we discovered that the reason that they don't like sweets is there's higher levels of a hormone called FGF21. And FGF21 is being developed as a fatty liver drug. So it's sort of like these GLP-1 derived drugs where people attach a lipid to it, or they make amino acid modifications so that the GLP-1s, these are much longer lasting hormones than the ones that are produced, endogenously. Similarly, people are developing FGF21 as a drug. But once we realized that the mice, and we think people with this disease, have high levels of FGF21 that's driven by a biochemical process that we discovered that's driven by a small molecule, we thought, oh, you know what? We can modify that small molecule and we'll be able to develop potentially an orally available compound that will induce the production of FGF21. We think that we will be able to develop a small molecule that will activate the liver's ability to make FGF21. And that could potentially be used as a treatment for fatty liver disease, as well as if you get high enough levels of FGF21, you don't like sweets.
Right.
And you're not attracted to alcohol. So that could be a first-in-class type molecule.
That's fascinating. GLP-1s are all the rage. In my clinic, I'm starting to recognize it as an increasing phenomenon, even amongst patients who are diagnosed with cancer. Do you view the two classes of drugs as being maybe synergistic?
Yeah, they've been tested. So in early phase trials, they've been tested and they synergize, just like GLP-1 synergizes with GIP. There are some biologics that have already combined GLP and GIP, and we think that there's going to be therapeutics that combine GLP-1s with FGF21s. Once again, our approach is a small molecule to get the person's liver to make more themselves.
And just, a thought, one of the things that we battle all the time in the cancer clinic is cancer-related cachexia, these wasting syndromes.
Right.
Is there any thought that there might be a subpopulation of patients who may benefit from the opposite, from actually having perhaps developed a penchant for sweets and for food in general? Would that be helpful in any other context?
Well, probably resistance training, is probably highly indicated with GLP-1s, right? Resistance training and walking as much as you possibly can would be, and probably taking leucine or something like that is potentially an approach to oppose cancer cachexia. We're in a food environment where most people have a pretty healthy appetite. One of the things about FGF21 that's interesting is that in some ways it's appetitive, so it makes you want to eat protein. It makes you want to eat protein and drink water and kind of eschew sweets. And so there's some interesting neuroscience to it.
One of the things that we always struggle with within our disease team, so I'm part of a team that focuses on kidney and bladder and prostate cancer, is the premise of using pharmacologic manipulation versus what you just alluded to a second ago, which is exercise manipulation, dietary modifications, and so forth. Clearly, this must be an area that you've thought a lot about. As we look forward to the next 10 or 15 years or so, would you say we should invest our efforts more in pharmacologic reagents to help mitigate diet, reduce appetite for sweets and so forth? Or should we be sort of pushing an agenda of increased exercise and dietary regulation?
I don't know about dietary regulation. That's not my area of expertise. I know that you can run an experiment in terms of looking at the cost of junk food and big gulp drinks and this kind of things. I do think that we do need to do something about the food environment. We do need to create shady, walkways for people that are safe and that have bathrooms. I think there are studies in Europe that showed that if you put public bathrooms out, in cities, you have more older people feeling comfortable to go for a stroll, right? So you do want to increase energy expenditure. You do want people to have better dietary choices. For sure, there's a role of ultra-processed food in overconsumption of kind of empty calories, but there's still a need for drugs. There's one of the thing about GLP-1s is that they allow a person to succeed in changing their diet because they hit a satiety point much earlier than they would have without the drug. And so they wanted to live in a world without so much food noise. And from what I understand, taking those shots allows people to essentially meet their goals, but probably combining with resistance training so that they maintain their skeletal muscle mass is going to be really important.
One of the interesting things in the GLP-1 literature to me is that beyond improving some of the endpoints that they're looking for in studies within diabetes or the cardiovascular space, for instance, what they're seeing is that there's improvements in overall survival.
Yes.
Do you anticipate the same for this FGF21 mechanism?
Well, it's too early for me to say. This is something that we only know about in mice, and in terms of our small molecule, I can't really get out over my skis on that one.
No, that's totally fair. Now, you've got a terrific, as you mentioned, small department, right, at the moment. Give me a sense of what some of your junior faculty are focused on.
I hired 3 people so far in diabetes and cancer metabolism. So Sarah Shuck is right at that interface. So she works on glyoxal modification. So we talked about hemoglobin A1C. There's a set of chemical reactions that will take reactive metabolites of glucose and lead to modification of proteins. And some of Sarah's work has shown that it can lead to modification of DNA and RNA and nucleotides. And so she's interested in determining whether these glyoxal modifications are not only a biomarker of various disease processes, but may also be a driver of mutagenesis in the development of type 2 diabetes and certain types of cancer. Yinfeng Deng, I recruited from UT Southwestern. She's working on uridine metabolism. So uridine is a nucleoside, and she's discovered some novel regulation between the pancreas and the liver and adipose that relates strongly to diabetes and may also relate to disturbances in cancer metabolism. Zhao Wang, I also recruited from UT Southwestern. He's probably one of the leading molecular cardiologists at City of Hope. And he's strongly interested in dysregulation of heart function, how that can be affected by cancer chemotherapy. And going forward, we think that there'll be very important cardiometabolic toxicology discoveries coming from Dr. Wang's lab.
Oh, that's very cool. So it sounds like there's good diversity within the group in terms of the areas of focus. I see a lot of really, really exciting things coming out of the group. This podcast has within its title, On the Edge of Breakthrough. And one of the things I always ask my guests here is, what does that mean to you, being on the edge of breakthrough?
It's just really exciting to be working with trainees and with colleagues and asking questions. Why is this fly male? Why is this patient population have a fatty liver despite no excess of adiposity? And it's a privilege really to work here and to be able to direct a small team of very dedicated researchers and to have the infrastructure that we have to answer important questions. So I want to encourage people to feel good about, institutions like the National Institutes of Health. We depend upon them as well as private foundations for support to do the research, hopefully to be able to develop new therapeutic modalities that will help people.
Well, I love your approach to research, and I look forward to hopefully maybe one day collaborating with you myself. There's a huge link between obesity and kidney cancer, so maybe after this we could put our heads together and collaborate, Charles.
Absolutely.
Thanks so much for joining us today.
Pleasure.
This podcast was produced in partnership with the Maze Media Lab.